From the tiniest building blocks of life to the complex genetic disorders that shape human health. Today, we journey through the fascinating world of biology, starting with the cell. In 1665, Robert Hooke observed cork under a microscope and coined the term cell. He used a compound microscope and saw tiny compartments resembling monastery cells. Later, Antony van Leeuwenhoek observed living cells in pond water. Prokaryotic cells have no nucleus like bacteria and archaea.
Eukaryotic cells have a nucleus present: plants, animals, fungi, and protists.
From single-celled organisms to complex multicellular life forms.
Life began as unicellular organisms 3.5 billion years ago. Through endosymbiosis and specialization, multicellular organisms evolved.
Humans share ancient ancestry with these early cells. Mitosis creates two identical diploid cells for growth and repair. Meiosis creates four unique haploid gametes with genetic variation. The cell cycle includes interphase, G1 growth, S-phase DNA synthesis, G2 preparation, followed by M-phase mitosis and cytokinesis. Prophase, chromosomes condense, spindle forms. Metaphase, chromosomes align. Anaphase, chromatids separate. Telophase, nuclear envelope reforms. Cytokinesis, cytoplasm divides. Plastids are double membrane organelles in plants: chloroplasts for photosynthesis, chromoplasts for pigments, leucoplasts for storage. Plant cells have rigid cellulose walls for protection. Animal cells lack walls, allowing flexibility and varied shapes.
Schleiden and Schwann established that all living things are made of cells, the basic unit of life.
However, this doesn't apply to viruses. They're cellular and need hosts to reproduce. Spermatogenesis creates sperm from spermatogonia through spermatocytes and spermatids to mature spermatozoa.
Oogenesis forms ova and includes polar body formation as byproducts.
Key terms: spermatogonia are diploid stem cells. Spermatids are haploid precursors. Primary and secondary polar bodies are meiotic division byproducts.
Recombinant DNA technology combines genetic material from different sources, like chitinase genes for antifungal properties or human genes in bacteria.
Lymphocytes include B cells for antibodies, T cells for cellular immunity, and NK cells that destroy infected cells.
Genetic disorders result from chromosomal abnormalities. Let's examine the major syndromes affecting human health.
Edwards syndrome, or trisomy 18, involves an extra chromosome 18. Symptoms include intellectual disability, heart defects, and distinctive physical features. Most cases are fatal within the first year.
Down syndrome is trisomy 21, the most common genetic cause of intellectual disability. Features include distinctive facial characteristics, heart defects, and increased Alzheimer's risk.
Patau syndrome, trisomy 13, causes severe intellectual disability, cleft lip and palate, and multiple organ defects. Survival is typically limited.
Turner syndrome affects females with a missing X chromosome, causing short stature, infertility, and heart problems.
Klinefelter syndrome in males involves an extra X chromosome, leading to infertility, tall stature, and feminine characteristics.
Cre-du-Chat syndrome results from chromosome 5 deletion, causing a distinctive cat-like cry and developmental delays.
Management focuses on supportive care. There are no complete cures, but quality of life can be improved through comprehensive medical support. From the first cell seen through a microscope to the frontiers of genetic engineering, the story of life is written in biology, and it's still unfolding.